I hope you're all doing well. Thank you all for coming. Instead of, I hope all of you can hear me. Yes? I hope all of you can see me. Because every time I go behind the podium, people say, Raju, get up, because they can't see me. So I'm going to use the stage here, and I want to make a few remarks before I bring the panel in. First of all, you see that there are three organizations here, and one of them that was established first is the National Foundation for Cancer Research, and you have heard earlier that it is going to celebrate its 50th anniversary next year. The second organization got started is the Asian Fund for Cancer Research Limited, and you'll hear more about that in a second. And the third one that started a couple of years ago is the Aim High Accelerator Fund. All of these three organizations have a common theme, but the glue for all of these three organizations is the head of each of those organizations, which is Suzanne Ba. And I'll say a little bit about Suzanne in a second. But each of these organizations is a not-for-profit organization. They depend upon people from around the world to recognize the mission of these three organizations and provide support for these organizations. And Suzanne manages how to deploy those funds, to the good of cancer research and cancer welfare all over the place. So first, as I mentioned, the organization that really got started with everything is the National Foundation for Cancer Research. And the leader of that organization for a very long period of time, more than 30 years, is our very good friend, Franklin Salisbury Jr. Franklin, please stand up. Franklin was the indomitable leader of this organization. He built it into the organization that is what it is today. And Franklin, we cannot thank you enough for all the things that you have done for the organization. We love you. Now, when Franklin, when he started the organization, he said he wanted to be able to change cancer. And the way that you'd be able to change cancer is to be able to promote, great researchers to be able to do great research. And their research discoveries are going to drive, how cancer is going to be treated in the future. And so he said, I want to be able to identify people from around the country who are doing great research or is beginning to do great research that have great potential, and I will support them. And that's what Franklin did. And the fruits of that effort clearly can be seen in many of the discoveries that have been made. Also, during that period of time, another way of recognizing the National Foundation for Cancer Research is by running the St. George Prize. And the St. George Prize always recognizes the greatest cancer researchers, around the world, and to honor them, in this event once a year. And you see the pictures of all of them around us, and some of them are here. And I am so thrilled that today, in advance, I'm going to congratulate Rakesh Jain, my great friend, collaborator, colleague, Rakesh, to be winning the St. George Prize. Rakesh, we're so proud of you. All right. So that was an effort. When Suzanne took over as the CEO of the National Foundation for Cancer Research, something changed. And that I think all of you see and recognize that Suzanne is a dynamo, right? She never stops, right? not only in terms of the efforts that she puts in, but the ideas that she has about how to be able to make cancer care a reality for everybody around the world. That was actually one of the reasons why she established the Asian Fund for Cancer Research, because NFCR was focused on the United States, and Suzanne wanted it to be focused on the world. And the way to be able to do this is to be able to create another independent organization that is focused and based in Hong Kong, and to be able to bring all of these things together and to be able to do a world of good. And you've actually seen some of those results today of some of the people from around the world coming up to receive recognition for the work that they've done. That's really fantastic. But that's just one part of what Suzanne does. But Suzanne, you may not recognize it, is absolutely impatient. She doesn't want to wait for things to happen 10 years from now or 15 years from now. She wants for them to happen now, today. And she was thinking about this. How can I change it? And said many times, and many of the people here in the audience who are involved in drug development know that the time period for the discovery to actual drug development could be 15, 20 years, some cases even longer. And Suzanne said, this is good, but I want to be able to do even better, to be able to accelerate this process. And that's how the idea for the Aim High Acceleration Fund, as the name implies, comes about. And these organizations are now trying to do things in a different way. The organization continues to provide great research support, and we have heard some of those fantastic scientists, so many of them members of the National Academy, sitting here on the podium. I was so glad to be able to see all of these people talking about their research, which is so exciting, right? And that you should be proud that you're supporting the research of all of these people, Suzanne. But in addition to that, she said, if we want to be able to bring cancer care to people around the world, what we gotta do is also to provide support. Identify young people who have new ideas, how they want to be able to take those ideas into the clinic, and to provide them support in many different ways. And one of the most important ways that you could provide support is to be able to provide money. And so Suzanne not only, put support from her own organizations, but she's a remarkable person in establishing networks of people, who are interested in cancer research and cancer drug development, bringing them together, making consortia, and putting money together and help companies to be able to do that, right? And what we're doing this afternoon is actually a part of that effort. And to say it's not just enough to be able to identify people, identify ideas, give them money, but also provide them support. And how do we provide that support? And you have heard from the great, from Wilson Sonsini. Wilson Sonsini was the corporate attorneys for my first company that I started in 1988 in California. So good to have you here and be part of this effort. Anyway, so that's part of the effort. And in the panel, we're going to bring, people, some who are very experienced, some, early stage entrepreneurs, and who are involved in companies. And we want them to share with us their experiences about building these companies, so that we can learn from them, and we could also get to know them and be able to help them as they put their companies together. But one last thing before I invite the people, and it's always wonderful to be able to have our friends and colleagues receive awards and so on. And so it's a particular pleasure for me today to be able to have not only Rakesh getting the prize later, but my very good friend Anna, receiving the first inaugural Beacon Award. Anna, you are a magnificent person. And I don't know how to describe you. You are a force of nature. And Anna, whatever she sets her mind to gets done. And she's a great friend. And I'm so proud that you won the award and sort of truly be the beacon for everybody. Thank you so much. All right. So thank you for indulging me in that little talk. And now I want to invite our panel members to come up here. All right. So the way that we thought that we would organize this panel is that we have a great group of people. And we will give each of them 5 minutes to tell us who they are and the company that they're representing and what they're doing, and also pose one idea or challenge, in building the companies that you are building. Okay, so we start with Paul, who is the Chief Scientific Officer of Oxford Drug Design.
Thank you. Okay. I'm also challenged with height, but I hope you can see me. So thank you for the NFCR for the invitation to present as part of this panel. I'm Paul Finn. I'm the Chief Scientific Officer of Oxford Drug Design. Oxford Drug Design is a spin-out company from the Department of Chemistry, University of Oxford, and we engage in two main activities, one of which is the development of computational chemistry tools for supporting drug discovery with a strong emphasis, but not exclusive emphasis, on machine learning. And then we apply those methodologies to our internal pipeline of drug discovery programs, which are focused on oncology. So just briefly about the platform, we have developed a wide range of tools over a significant period of time, dating right back to the company's academic origins from Oxford. I'll just talk about two of those very briefly to give you a flavor. One is the development of novel and proprietary molecular representations so that we can search very large cheminformatics databases very quickly for the identification of novel active compounds. And that data, together with data from other sources, goes into our machine learning platform. We like to develop explainable models instead of the sort of neural networks that tend to be sort of more black box in their approach. And we like that because we think that leads to rapid and more effective decision making and better communication between the project scientists. We've also developed a significant expertise in the aminoacyl tRNA synthetase family of enzymes, which Zhang Lei introduced us to just before lunch. In addition to their well-known role in protein synthesis, during the course of evolution, these enzymes have acquired many additional so-called non-canonical functions, which are involved in many important processes in the cell. And we believe that this is an underexploited family of therapeutic targets, which we are very interested in developing further. One example of that comes from our work on the lucile member of the synthetase family, which we're exploring as a novel approach to mTORC1 modulation. I don't need to say anything about mTORC1 to this audience. It's clearly a key regulator of cell growth and proliferation. And the involvement of LARS1 comes through the fact that mTORC1 is activated by amino acids, and LARS1, the tRNA synthetase, plays a role in that as acting as the intracellular sensor for the leucine concentration, and by inhibiting that, we have a novel way of modulating mTORC1 activation. The two current approaches that are most widely used are through rapamycin and rapalogs, which through its mode of action only inhibits some substrates of mTORC1, leaving others relatively untouched, and that is related to limitations of efficacy of those approaches. The other approach that's been widely studied is developing kinase inhibitors to the mTOR kinase. These are able to inhibit all substrates. However, they have the unwanted effect of also being inhibitors of mTORC2, which leads to dysregulation in metabolic functions such as insulin sensitivity. The LARS-1 approach can overcome those two deficiencies, we believe, and by developing these inhibitors of leucine sensing, we will be able to develop molecules that, like the kinase inhibitors, affect all substrates of mTORC1 and can modulate them all, but unlike the kinase inhibitors, will be selective for mTORC1 because mTORC2 is not activated by amino acids, but by completely different mechanisms. This project is progressing well. We're very lucky to have access to our own proprietary structural biology to help the chemistry optimization. We have compounds which have demonstrated selective mTORC1 inhibition and compounds that are active against a range of cancer cell lines with clear SAR to guide optimization, and we're progressing this program towards in vivo evaluation. The company is small, but we have experience from both large pharma, biotech, and academic spheres, as well as commercial and financial aspects of the company. And we've made significant progress since our last funding round in 2019. We're currently engaged in fundraising with the aim of raising $12 million, with the key objectives of accelerating this LARS-1 program that I just told you about, continuing to expand the AI platform to support those programs, and initiate additional programs in this area of the non-canonical functions of the amino acid tRNA synthetase, which we believe offers a large number of opportunities in multiple therapeutic areas, but including oncology. If I have a challenge, it's fundraising. That's the biggest challenge for what we have to do. And with that, thank you for your attention.
Thank you so much. Thank you. And we have Pritesh Gandhi, who is the CEO of TellBio, and you heard about that a little bit this morning, and he'll tell more.
That's right. Good afternoon, everybody. Thank you, Sujwan and NFCR, for inviting me. This is an honor and a privilege. My name is Pritesh Gandhi. I'm the CEO of TellBio. We're a spin-out from Mass General Hospital. So just in terms of background and context, as you think about what the unmet medical need is in cancer, we, of course, talked about it all morning and all day today. But just this year, more than 10 million patients will die of cancer. And if you take a look at the major reason for the death of cancer patients, it's metastasis. More than 90% of the patients will die because of cancer metastasis. So what we really want to do, of course, is we want to try and develop a company that is going to be able to detect cancer metastasis earlier through a liquid biopsy, specifically by looking at circulating tumor cells, and learning the biology of these circulating tumor cells so that we can target and guide therapy against these circulating tumor cells so that we can have an impact on the outcome of patients with cancer metastasis. Today, there's been a ton of progress that's been made in terms of developing drugs for the metastatic disease. However, there are no drugs that have been approved for targeting cancer metastasis, specifically the metastatic process. So who are we? We are a spin-out from Mass General Hospital, specifically from Daniel Haber's lab. You heard Dan speak earlier today. We have proprietary technology. We're an integrated diagnostics and therapeutics company focused on circulating tumor cells. And of course, these circulating tumor cells are cells that dislodge from the primary tumor. They end up surviving in circulation, and then they potentially deposit in secondary sites to cause cancer metastasis. What we really want to do is to be able to identify these CTCs, or circulating tumor cells, through our proprietary technology, and then really learn the biology of these circulating tumor cells and target therapy. We know that circulating tumor cells are prognostic. These are data that are from MGH. On the left-hand side is the Kaplan-Meier curve in breast cancer. On the right-hand side is in prostate cancer. And you can see specifically that the more circulating tumor cells there are, the worse the outcome. And this has been seen in a variety of different tumors. Our technology is able to identify circulating tumor cells agnostic of the solid tumor. And we're also starting to do some work with the Dana-Farber in multiple myeloma as well. So the question now is, it's hard to tell a patient that you have circulating tumor cells and you may have a worse prognosis. The question is, what are you going to do about that from a treatment perspective? And so what we're doing is really learning the biology of these CTCs and guiding therapy. So ultimately, where do we want to go? We want to go in a direction where we can detect cancer metastasis early, we can monitor therapeutic efficacy, and then we can guide therapy against circulating tumor cells. So our operational plan is to be truly end-to-end, in that we would collect a blood sample, 5 to 10 mils of blood. We would basically look for circulating tumor cells in these blood samples. We would molecular profile these circulating tumor cells and then enrich the patient population. Guide therapy against those circulating tumor cells, would monitor the efficacy of that therapeutic with the diagnostic, and then with the aim of enhancing and optimizing outcome for patients with cancer metastasis. Thank you.
Thank you. Next is Vlad Hogenhuis. Vlad is the CEO of Chimera Bioengineering in California.
Thank you. First, I'd like to thank the NFCR for inviting us. It's truly a privilege to be here and represent my company, and also for the Aim High Accelerator Fund to invest in our company. Their work and due diligence really brought in a lot of new investors into the company that have allowed us to move forward. A little bit about Chimera Bioengineering. We aspire to treat cancer with dignity and humanity. I think everybody in this room agrees with that. Our founders really had a very personal brush with cancer, either themselves or their family, and really started to come together 7 years ago in founding the Chimera Bio company. I've been now CEO for a year, came on board in November. It's been really fantastic to work with a small company that has this vision of addressing cancer. Specifically, our mission is to address the solid cancers, and we're doing that through, in particular, cell therapies. Autologous CAR-T is our first stop. Our specific approach is that we have a technology that we call GOLD, that is a transcriptional modification that governs the switch on which lymphocytes release cytokines. We've discovered that switch in our labs, we've patented it, and now we use that in all of our therapies to arm CAR-Ts conditionally with very powerful immune modulator cytokines such as IL-12, because we believe that arming cytokines holds the promise of addressing the tumor microenvironment through which many of these cancers protect themselves, and where unfortunately many cell therapies efficacy has proven elusive. By the way, we call it GOLD, not because it would be anything financial, but it is an AU-rich domain that is post-transcriptional, and AU is gold in the periodic table, so we've now labeled this GOLD technology. In terms of the company, to our knowledge, we're the only company that has really identified this switch and is able to deliver the efficacy of a cell therapy that's augmented with a payload. And we do that with a high therapeutic index, that we can really turn it on completely when it needs to be turned on in the tumor and turn it off completely when it's outside of the tumor. And some of our competitors claim that, but they cannot really get to that zero-level expression that the lymphocytes have that protect the cytokines that they're expressing. So this is really kind of important for payloads such as interleukin-12, that if they're not appropriately used, can use significant mortality and morbidity. We also think that this approach is better than either naked cytokine or masked cytokines. Unfortunately, while these technologies are promising, there's always a background level of leakage that leads to AEs that we're trying to avoid. Now, we've really been building hard with the funds that we received from Aim High in building a pipeline. CBio7 is our front runner. It's an autologous CAR T therapy targeted against TNMUC1. We heard about this morning about several researchers focusing on TNMUC1 as a target. It is a tumor-restricted antigen with very little expression in healthy or no expression in healthy tissue. Our approach is to do a basket trial around December of next year at Roswell Park Center in Buffalo, New York, with Rainier Brenchen's team, where we're going to be looking at the number of TNMUC1 positive trials and evaluate the efficacy in our and safety in our phase 1b trial. We have a fast follower compound, which is CBio7 Allo, that has some promising proof of concept data. And you can see the rest how we're building out the platform with one compound, one CAR T targeting DLL3 that's augmented with a double payload, as well as an undisclosed AML target. So really building out a pipeline that's focused on the augmenting CAR Ts with a payload. We're also very busy here in actually getting to this trial. So the road ahead for us is to get to the IST trial and get a readout of that. Number two, manufacturing GMP supply so we can do all of the preclinical experiments that lead us to the IND, as well as furtherly developing our pipeline. And this will really allow us to be kind of a one-stop shop for not only ourselves, but our partners, because we believe that our technology works with a whole host of engineered cells, not just autologous therapies. And this is really, I think, to your question, Raju, what is our biggest challenge? That's fundraising. And so we are kind of at this time raising a $50 million Series B round. This will pay for the IST trial, the manufacturing, as well as our company IND. And will advance our pipeline. And we believe that with these milestones in hand, we'll be able to create material value to our investors and set us up well for a Series C down the road. And again, really grateful for Aim High to be one of our investors. And if you'd like to talk more about that in the break, we could talk about this, or you want to do any collaboration project on with an armed CAR T for a target of choice, certainly very interested in exploring that. Thank you for your attention.
Right. Thank you very much. Reflecting the international scope of the portfolio companies, we have John Luke, John is the CEO of Arbele. Please.
Thank you. Good afternoon. I'm very pleased, Sujan, inviting me to be here as a portfolio company on behalf of the Blaze Award winner. We are the first Blaze Award winner. I have to give the credit to our Chief Medical Officer. Actually, he pitched for that and won $300,000. Okay. Next time when I pitch, I hope you give me more money. This is very special for Hong Kong because, you know, the last clinical award was won by Dr. Dennis Low for his discovery on the fetal DNA in the bloodstream. And also my mentor, when I did my postdoc fellow on Asian molecule, he also won the Alaska Award this year. So it was a kind of a very, very exciting year for me. So my company is Arbele with a specific aim to develop targeted immunotherapeutics to treat advanced GI cancer. Because when I was in PHARMA before, the company not interested in liver cancer, esophageal cancer, because there's no market for the big pharma, because in Asia, the patient cannot afford to pay such expensive drugs. So that's why my company is really aimed to develop new drugs for the GI cancers. So needless to say, GI cancer, according to the WHO report 2 years ago, about 5 million new cases per year, and account for almost 1/3 of all the cancer deaths, 3.3 million deaths. So I believe the deaths are increasing year by year because of the late detection or late diagnosis of the GI cancer. And as we all know, the fecal blood test is one of the very standard test, but the non-sensitivity and also the false positive is very high. So the gold standard for the definitive diagnosis is by colonoscopy or by gastric endoscopy. But these procedures are very invasive and also are very expensive. Okay. So our company is really looking for not only drugs, but also intervention platform to early detect GI cancers. So our company focuses on 3 pipeline strategies. One is targeting pathway-specific markers using monoclonal antibodies, antibody drug conjugate, bispecific, BiTE antibody, and also a tri-specific antibody platform. And the second pipeline strategy is focused on precision medicines. So we develop our own proprietary IVD platform to look at the samples and generate a score for selecting the patient. So we just want to treat the patient that express the marker for our targeted therapies. And also now it's also very interesting. Last month we also found out that we can take a drop of blood and test the marker in the blood. We can separate the patient with polyps versus the normal healthy individuals. So actually it has a huge market potential. That means that we can screen those at-risk subjects at 50-year-old and above by doing a blood test. And then those are positive, we can subject them to a colonoscopy. So why I say that? Because in our early screening, we invited our board of director and also the lab mate to do the blood test. And one of the board director, actually he has a very high level of the marker, and then we see possibly a lab error, so we test again, and again the marker is also very high. So we advise him to go to the doctor to see something wrong, and he actually did a colonoscopy and found out there's several polyps, and then we removed the polyps immediately. So I think that would be a great potential. So right now we are collaborating with Oxford, trying to make it like a POC test, so everyone will be available over the counter. And the third pipeline strategies, we are looking at the niche area, such as orphan disease, cholangiocarcinoma, gastric cancer in the US is also considered as a orphan indications. So we try to leverage the chimeric antigen receptor. We are using the non-viral base and also using the allogeneic NK platform, trying to deliver the CAR cells to the patients. So this is our pipeline. Our leading molecules, bispecific antibody BiTE, already going to phase 1 this year. And we successfully dosed the first patient, and now we are moving to the second cohort. So far the data, no data, that means no finding, that means good for us. So that means no adverse side effect. And the second one is the conventional CAR-T. So we are going to IIT early next year. And then the ARB101 and ARB203, actually they are moving much faster. So we believe these 2 molecules will go to IND enabling stage next year. Okay. And as I said, we also are doing a diagnostic. So we have the TIP17C is an IVD automated IHC staining platform. So we can put our platform into the Roche Ventana platform, or also put into one of the Asian-made staining platform. And the TIP01B is the blood test we are developing now, going to hopefully we'll move to the POCT platform and trying to commercialize as soon as possible. So the company roadmap, we started company in Hong Kong in 2016. So for those 6 years, we have been encountering a lot of hurdles, such as money, talents, and also the environment. I was told the best area to start a startup is in Bay Area in Boston. So in Hong Kong, it's a desert. So there's a lot of money, but only for finance, okay, for real estate. But for biotech, it's like a desert. So it's very difficult for us to raise money. And we really appreciate Suzhong give us 300,000. Hopefully we'll be more. I repeatedly say that. So all this jargon, MME, and then a few years ago, we formed a joint venture with Avalon Global Care, trying to develop CD19, CD22 dual CAR. And then we had a PharmD meeting with FDA, so we plan to expand the trial to US, one in the New York State, one in Penn State, and one in California State, starting the phase 1B to 2A study next year. So another challenge is the team is very important for a startup. So we are very lucky and fortunate. Our core team mainly come from industry. I'm kind of a hybrid. I have 18 years in academia. I thought I'm going to retire as a tenure professor, and then suddenly Roche had come to me and locked my door. John, I want you. Please come to lead our oncology discovery. So I have been in industry for almost 10 years, and Dennis is our chief medical officer. He has been working at Roche, Genentech, BMS, and Sanofi for over 20 years. And our CTO, Anthony, he is a Cambridge Ph.D. graduate, and he has been working in cell therapy for 15 years. And our co-founder, Alec Huang, also has been working in industry for over 25 years. And also we have some young generation. I'm looking for my successor. Hopefully we have our successor on track, including the young lady, Diana, also from working at J&J and Takeda, and Stella focusing on translational medicine, and Tony working on discovery. So this is our key partnership. As you can see, after our first patient dose successfully, we have a bowling competitions. So all the young people will go to bowling competition, and as you can see, we have a AFCR award we have to acknowledge. So we get most of the funding, 1/3 of the funding from the government. They are non-diluted. And I'm glad to tell AFCR the money you invest have been triple the valuation already. So we have 2 pharma approaching us for potential out-licensing for our lead compound. So I think I keep in 5 minutes. Thank you so much.
Thank you so much. Yeah. So I recently had an opportunity to take a look at a fantastic photograph taken by the James Webb Telescope. And this picture is of the universe, a part of the universe where there is this very large region of high levels of dust and gas, which is the cauldron in which stars are born. And when I saw that picture, the person that I was reminded of is Ken Malone. Ken takes this kind of material and converts them into fantastic companies. And he's a great entrepreneur. And Ken is from Baltimore. And he's the CEO of a big company called Early Charm. And maybe he'll tell us a little bit about Early Charm and one of the portfolio companies, Rasio. Ken.
I don't think I can follow that, Raju. Oh, goodness. I promised the other panelists in our meeting a little earlier this week as we were planning this that if any of them ran over the 5 minutes, I would give them part of my time. So I'm now done. Look, Rasio Therapeutics is actually a kind of a special company. And I think that if I can figure out how to push that button there, I can explain that it's actually a joint venture between an existing company that I had, Silks Bio, and I'll tell you a little bit about Silks Bio here in a second. But a fairly excited person met me and discovered what Silks Bio did and said, we need to create a joint venture, not to solve any one drug for one oncology application, but for every. And we're going to solve all therapeutics for every possible cancer. And I said, okay, that sounds like a good goal. Let's do that. And so we created this joint venture. And let me tell you a little bit about what SilksBio does first. So much like a mechanical engineer sits in front of his computer and models a device, and then looks at that device, and that device, the modeling was done based on physics, right? We use physics to do the same type of modeling of a protein, except maybe instead of looking at the force factors and the pressures and so forth that you see from a mechanical engineer's design, we're actually seeing the hot spots of where chemicals would best fit and attach and bind to a protein, right? But we're doing it by physics, not by AI. There's no guesswork in what we're doing. We're actually doing very accurate calculations. Now, people have been doing that for almost 30 years. What we're doing is a lot better for several reasons. Number one is you can do calculations a whole lot faster today than you could do 30 years ago. So the algorithms you can run are a lot more accurate today. And the second is we've learned quite a bit about how to run those models much, much, much faster. Because honestly, if you just use quantum mechanics and you had an infinite amount of time, you could be exactly precise in every molecule that you designed. So all of computational drug design is a compromise between quantum mechanics and its exact nature and the ability to come up with something that's good enough. So SilksBio we sell software to all the large pharma companies. Everybody buys this software today. It's very well understood and used, right? And it's used and validated and understood in all these different proteins and so many more, and I'm not going to spend a lot of time talking about that, right? What I will tell you is that we've built this better than AI platform, but SilksBio is just selling software. And people don't pay a lot for that at the big pharma companies, and they don't pay for it for a couple of reasons. One is the pharma company doesn't want to solve every single oncology drug this could possibly ever be solved for. They just have one target they want to work on. Well, the beauty of what we do is we're able to do very, very rapid calculations across a very large portfolio. And so Jean saw that and said, we need to do something much, much bigger. And she brought not just the money, which is nice, she brought also all of the knowledge that we didn't have in terms of how do you look for targets and what do you work with and who are the experts in the field for those targets, right? We didn't know any of those things, we were just selling software, right? On top of that, she brought a great network of investors. Very thankful that she brought in Blue Dot. And together, we've created what is now Rasio Therapeutics. And I'm sure it says something really great on that slide, and there's a little box up there that says it, and I can't read any of it, so I'm going to make it up and say as I go along, right? What have we done, right? We've run a whole portfolio of proteins, right? We're able to very rapidly develop molecules, and we're working on some of those molecules moving forward, but what's even more interesting is we've been able to develop a whole portfolio of these proteins that you don't want to hit, right? Because it's actually not all that hard to come up with a molecule that's going to stick well to a protein. It's really hard to develop that molecule that's going to stick well to the protein and not hit 20 other things that you don't want it to hit. Right? And what do we need? Well, I think we need money. No, seriously, we don't need money. You know what we need? We need enough success and enough proof of what's working that money finds you. By the way, that's the way you raise money. And anybody who tells you raise money by just knocking on doors and begging for it is wrong. You build the right thing, and the money finds you. Thanks.
Thank you. And now we have María Varela. María, you met earlier, and last year she competed in the women's venture competition and succeeded. And María is from Uruguay and Argentina. And Suzanne tells me that she's the youngest CEO that she has known. And we're so proud to have you here.
Thank you, Raju. Thank you, Suzanne. Hello, everyone. It's a pleasure. It's an honor to be here, where everyone is trying to help patients in one way or another. And so it's my work to let you know a little bit about how we are doing or how we are trying to help patients from ARDAN PHARMA, which is a spin-off from Argentina and Uruguay, as you will find out later. So basically, what we are trying to do is to present a novel target to modulate inflammation in cancer context. So this target is able to control inflammasome activation, and we want to develop small molecules in order to target this molecule. Up to now, we have in vitro and in vivo proof of concept with this small molecule we call boritinib across different types of tumors. This investigation led us to file 3 patents, and we also are working on a molecular signature in order to have translational studies that could develop biomarkers to assess clinical studies. So, meet TORID. It's a short name for transmembrane protein 176B. Basically, TORID works as a checkpoint of the innate immune system. It impairs inflammation. It's a negative regulator of the NLP3 inflammasome, and therefore promoting an immunosuppressive environment. Now, interesting, metastatic melanoma patients who were receiving anti-PD-1 immunotherapy evidence a correlation between TORID overexpression and bad outcome to this treatment, suggesting TORID as a resistance factor to anti-PD-1 immunotherapy. Now, there's a big step between identifying a target and actually having a target that you can develop a therapy around it. And this is basically all our efforts since we started our company. The guidelines summarize it very well, and you need to have these 4 key pieces of the puzzle in order to work it right. It's causality, the safety, the feasibility of the target, and also the strategy. So basically, we have assessed TORID role in cancer immunotherapy, the link to the disease, also with several experimental models, as well as the most important primary cell lines. We also assess the safety of the target, the safety of TORID being itself with, does it have any healthy warnings that we should worry about? And interestingly, apart from the experimental assays, which I found the knockout for TORID mice the most safest ones that we can get right now, we also investigate that there's a 30% of the population that has polymorphisms of loss of function for TORID and had never expressed any health compromise or any other issue at all. So also the feasibility of the target, we have developed bioassays around it to understand how it works, if we can inhibit the target or the other way around. That took us also to have this tool compound and also the start working on biomarker development. And last but not least, the strategy. The unmet medical need of cancer immunotherapy resistance is something that we already know, but also finding an intellectual property strategy in which we can work on. So summing all this, we came up to develop TORID inhibitors. So boritinib is a small molecule that by blocking TORID, it is unleashing 2 parallel mechanisms. One is triggering immune system modulation, and the other one is directly promoting the malignant cell death in an inflammasome-dependent manner. So basically, this is our competitive advantage and what will give us a business strategy to move on. So a little bit about our proof of concept. When we administered boritinib into in vivo mice models of chronic lymphocytic leukemia animals, we increased survival as we are promoting the leukocyte death. Similarly, in T-cell lymphoma mice models, we increase survival. And the interesting thing is that when we knock out TORID, we lose this effect, somehow proving the on-target efficacy. What is more important is that when we combine boritinib with anti-PD-1 immunotherapy, we can increase overall survival in preclinical models of colon, lung cancer, and melanoma by 40%. So basically, upon all this, we created our portfolio of assets. Up to now, boritinib is our most advanced asset that it is somehow proving viable properties that help us to engage boritinib in a lead optimization program that will help us and enable us to file our first composition of matter patent. So I will be enough with the technology. I want to tell you a little bit about our story right now. As Raju mentioned, I'm from Uruguay, and we created ARDAN PHARMA as a spin-off from academic institutions, particularly the Instituto Pasteur of Montevideo. We were invested by a fund in our neighbor country, Argentina. in order to have a better formation on the business aspects. I moved to Argentina. I was living for almost 3 years now, and I currently travel from one country to another one. But of course, we want to bring this technology to the US. We started by creating and establishing a holding corporation at Delaware. And well, our initial capital I already mentioned, but also what fuels our technology is also grants, as we are at the early stage of research. We have an NIH grant, we have an international, we have a national from the local government of Uruguay grant also supporting our technology. And last but not least, the research has been made in laboratories that were labeled by Centers of Excellence by the FOSIS or the Federation of Clinical Society Immunology. So I want to present my co-founders. Both Drs. Marcelo Gil and Pablo Peso are immunologists. They are principal investigators at the Instituto Pasteur of Montevideo, which is where most of our technology and our research was developed. I have a scientific background. I don't think I mentioned that. I am a biochemist by training, and when I was under my postgrad studies on biotechnology, I met them and I jumped to the business side. And I think I wouldn't have done it if I couldn't meet with these amazing people that were willing to work with us, especially giving us very advice on the business side of the life science cycle on the business development, specifically by William Mann and Dr. Nicolas Toniali. Also from the drug development expertise, we count very much on Silvina Garcia Rubio. And on the most clinical investigations, though we have quite a few steps to go, we count very much on Mercedes Lassus and Adrian Rubstein. So that's all for now, and this is just to invite you to go to Uruguay. Okay, so thank you.
Thank you so much. So I want to have the discussion in a slightly different way than we usually do, because the audience has a fantastic amount of people who have great amount of experience. And I'm going to actually cold call on people to make some comments about the first aspect that we talked about, which is fundraising and financing. So I want to ask Ron DiPino, my great friend, former St. George winner, and past president of MD Anderson Cancer Hospital, serial entrepreneur, to get up and say a little bit about this fundraising issue. Ron.
Thank you, Raju. I'll put it in a little bit of perspective. So there have been 4 downturns in the last 20 years, 2002, 2008, 2016, and this past year. And in each of those, biotech survived. And what made the companies that survived had great science and great people. And they also learned how to utilize the resources, the limited resources that they had. In each of those situations, it really helped to try to diversify your revenue stream. With the recognition, it depends on where you are. If you're early stage, you're actually in a better situation, Series A and so on. If you're later than that or even closer to crossover, these are extremely difficult times because the IPO market isn't open. But I think the common theme that I've seen is that you have a horizon of about 10 to 20 months before you come out of this situation and the markets start to open up and crossovers start to emerge and so on. But in the interim, it's very important to engage your investors, maintain your relationships with venture capital, who need to put their resources to work and have to fill their pipeline for the future. But very importantly, right now, I'd say the biggest source of cash is large pharma, which has done extremely well. And we're seeing a huge amount of business development deals. So I think one of the most important things is to really engage large pharma. And they too have to maintain their pipelines and so on. And they recognize that they're in a pretty good buying position. And so they think that's a huge opportunity as well. And I'd say the third axis is our grants. So what NFCR is doing or other organizations like CPRIT or the NIH can provide critical funds, especially for the early stage companies. So I think that I think for me, from having gone through these things a number of times, it's really engaging all mechanisms to generate revenue while maintaining the one thing that you have control over, which is your expenses, and being very thoughtful about prioritization.
Thank you, Ron. So, Ken, you talked about a new type of model, and you suggest that the companies keep their nose to the grind, and investors would come looking for you. Tell us more.
It's easy, Raju. I don't know why everybody complained. No, seriously. Look, several things. One is venture capital has a very narrow window that it's interested in, right? There's always specific things that they want to see. And if you're not that, you don't have as much of a chance there. But there are other opportunities, right? I think partnerships with large pharma, partnerships with other organizations that are looking to accomplish similar goals is a really critical area to be looking at, right? And the other piece of it is, we have this interesting model, right? If you have a single molecule therapeutic, you really only have one pathway, right? You have to raise a big bucket of money or it dies, right? But if you have a platform, you've got different opportunities. And several of the presentations here today were platforms, and those offer opportunities for different types of partnerships, different types of things like contract research and other ways of getting money. Sometimes it's just about surviving long enough to make it work.
Where's Beverly? Beverly? Do you agree with what Ken is saying?
She looks too smart. She's not going
Beverly
to.
Lou.
Yeah, I was trying to hide, by the way. Thanks, Raju. Well, who can follow after Ron and Ken, right? Well, from the VC investor, I'm going to try very hard to think of something else. I'm in wild agreement, by the way, but trying to be value add here. I think one thing what Ken said resonates in particular is about VC investors has something particularly they're looking for. So that brings me to this art of storytelling. So this is not something I've learned in grad school or my postdoc. This is something I learned when I was at McKinsey, trying to sell my project or idea to pharma CEOs. This is something I learned when I tried to get funding when I was at Amgen, running the US portfolio for a particular strategy. So this is extremely important. It is an art. You don't need to change. If a particular venture, what they're looking for has nothing to do with your technology, forget it. But oftentimes what I find is, as a VC investor, I have to look really hard for that shining piece in the technology, rather than it was presented sort of front and center to me. So that's one thing. Another thing, that's what Ron said, being creative in this type of environment. Ron is extremely successful and smart about that. We often have conversation about it. Another thing, though, along that same line is to really think through how to streamline. What is your next milestone that's going to trigger your fundraising? And try to exert the minimum resources and effort to reach that milestone. Of course, you want some buffer, but meaning you want to buckle down and survive this rough period of this 10 to 20 months that Ron just referenced. So those are the two things I struggle really hard to come up with just right now. Thanks.
Thank you. Vlad, you talked about your current efforts to raise money, and so how do you find the atmosphere, and what are the things that resonate with your investors?
Yeah, so I found there's a profound change that happened if you compare first and second quarter of this year to the third quarter that we're living now. I felt, that lots of VCs were actually staying and keeping their money, if you will, powder dry, and prioritizing follow-on investments in their initial venture, but very little investing in new ventures. But I think what you also see is that there is also an incredible selective factor of picking the top of the litter in investing in new money. I think what our company needed to realize that, I think what Beverly said, we really needed to kind of change a story from a Ph.D. thesis to something that a general investor could understand and could hit the, I would think, desires or priorities of different funds. So we repositioned to do less science, more kind of connecting, more storytelling, and more, I think to what Beverly said, pointing out the milestones that we're going to hit with the budget that we're looking for, and what would this do for an investor kind of going forward. So with that, we've seen, I think, a significant increase in interest starting from Labor Day. People came back. There was renewed optimism. The XBI had reached the nadir, and so there's a lot more kind of interest. What's important in this time, of difficulty is really to focus on bolstering your areas of validation. Can you validate yourself through a partnership with a top institute? Can you validate yourself through publishing your research? Can you validate yourself by pursuing non-dilutive grants that help you kind of build more research? And these are kind of areas of funding that are really merit-based and are potentially less fickle than finding the right VC at the right time. And I think that will allow you to continue to deliver and wait for the upswing to happen, because I see the upswing will happen, but it's a matter of weathering through this difficult period.
So I want to ask Catherine here. Catherine is an attorney with a large law firm, helps a lot of companies with many corporate affairs, but especially financing and bringing money in. Do you have advice for our people, Catherine, about what are the kinds of things that they should be doing in raising money?
Thank you, Raju. Pleasure meeting you all. I think Ron mentioned an important point earlier. I want to elaborate a little bit more. The federal grant, NIH, NSF, DARPA, and some other federal grants. In the last few months, federal government has passed a lot of the new legislations. For example, the Inflation Reduction Act, right? The name doesn't seem to be connected to biotech, but if you look closely, there are a lot of research grants directed at research institutions, but also private companies that you can apply. So follow, the federal grant government website and really look into that pocket of money. And then another place I don't think anybody mentioned, still international money, foreign investors. CFIUS is not as scary as people think, because if you look at the CFIUS 10-year report, even though people think CFIUS really deters foreign investments, but if you look at the CFIUS 10 years report, the vast majority of the deals don't get CFIUS scrutiny. The vast majority of the deals are approved by CFIUS. So foreign money is still there, and there's a lot of them. If you look at the statistics, a large percentage of US biotech research, including fundamental and translational research, traditionally was funded by foreign investors, and even more increasingly so. So don't be deterred by CFIUS and export control. Find ways around it.
That's great. I want to just move this slightly a different direction, but it's related. And one aspect of the financing, it's always wonderful companies to be able to have non-dilutive financing, and all of you have thought about some of those ideas. And one of those non-dilutive financings is to be able to work with other biotech companies or pharmaceutical companies to be able to have relationships not only to be able to provide financial support for the company, but to be able to have the intellectual input from them. I'd love to hear your ideas about that. Pritesh.
Yeah, thank you. That's a great question in terms of non-dilutive financing. I mean, for a company as nascent as ours, TellBio is, 2 years, we are an integrated diagnostics and therapeutics company. And we're lucky because of the diagnostic piece that is ready for research use only. We have now implemented and deployed the technology in a variety of different settings and actually getting revenue, which is generally unheard of for a company that's 2 years old. And so now we have deployed the technology within academic centers to advance translational research projects. We have integrated the technology within clinical trials to get earlier read of efficacy in terms of potentially reducing circulating tumor cells. And we've also worked closely with commercial laboratories, not only in the United States, but also in China, where we have now deployed the technology within commercial labs so as to be used as a research tool only and are generating revenue. So it is a way of getting non-dilutive financing, which is really helpful because it really fuels our R&D costs on the therapeutic side.
Paul, you have comments about this?
Yes, I agree with that, and I also, it's related to the point about having a diversification to give you a bit more robustness. We have in Oxford Drug Design, I think, been quite good at raising non-dilutive funding from a variety of sources, mostly in the EU and the UK, of course. But we've also had non-dilutive funding from US-based organisations as well, and that's been a lifeline at times in the company's history. And again, because of our computational technology, we've been able to, when times have got tough, to engage with other companies and provide sort of services and so on and so forth. That again has been very, very useful sort of during those difficult periods. So I think all of those approaches are really helpful during those periods so that you're ready for when things turn up again, that you can pick up and make progress.
I'm going to put another person on the spot here. Chikao who? Chikao, you are the CEO of a couple of companies that are developing new wonderful products that have been thinking about these types of relationships. Can you share with the people your perspectives?
Thank you, Raju. So I think there's a very simple saying, is time is money. And if we don't have enough funding, a lot of program has to be delayed. Right? That's just simple, conventional wisdom. So I think whatever the resources we have to constantly prioritize that. And so pharma development is not just reaching the next milestone. And I think pharma development, every day, all of us sitting in this room, we have to make a very tough decision. And I always tell my kids, whatever that he made a decision for you, you are going to suffer tomorrow, right? So the decision we make today during the downturn, it will change. Can we really harvest when the economy becomes better, right? So every single decision we make today, we have to figure out when the situation changes, does that the decision still hold true? So I think to make a decision, usually if you have a lot of funding, you make a decision for today. If you don't have enough funding during very tough time, you probably try to make a decision for tomorrow. Thank you.
Great. So María, your technology is completely new. The inflammasome is very exciting and it has potential applications in many areas. And I presume that your company cannot develop all of those products. You've been thinking about relationships with the biotechs and pharmas?
Yeah, but step by step. First, validating the technology in one indication, it seems the most reasonable. But of course, exploring potential partnerships after, for example, clinical proof of concept that gives you enough, that puts you in a place when you can make some deals with a licensor technology or something, and you could use that upfront payments in order to keep on research and maybe explore other indications. And of course, seeking partnerships in that way would make the most sense.
So John, your product is getting into phase 1, and so you must be very excited about that. You've also been thinking about non-dilutive financing and working with pharmaceutical companies or biotech companies?
Yes, I do. I think for us, my experience applying for government funding is as a backup. You don't get the money right next week, right? Usually the SBIR or NIH grant usually only several times a year. You have to prepare half year, you don't have money in your pocket. So you have to find something else. So either from your friends or from your wife or from whatever people. Yeah. But don't sell your house. This is what I learned to fund your company. And also I agree with Beverly. Setting the milestones is really important. And also you have to calculate how much resources, what kind of talents you need. Make sure whatever you promise to the investor, you can deliver. Okay, if you cannot deliver, you have to explain why. Otherwise, actually, I feel like a babysitting for the VC because your question is very difficult compared to the investors to answer. So, so I think keep a very good relationship with the investors and also with Sujang, very important.
Yes.
So hi, I'm Michael Saugeller from NCI. So I want to when you're talking about non-dilutive support, I want to put a plug in for partnerships with the National Institutes of Health. We're mostly known for the direct funding that we provide, but it's important that we work every year with hundreds of companies all the way from startups to public companies. These are partnerships that are typically cost and effort sharing. They're not competitive processes. They're, if we like the technology and we can find a scientist or a clinician interested in working with you, then you're in. We're also the largest clinical investigation center in the world. So if you have first-in-human trials, we run more of those than anybody, and we typically either fully fund or provide a lot of support. And finally, I would look, while you're looking to the federal government, look to the entire federal lab consortia, which are 80 labs around the country, which do what we do. So for example, DOD, the Congressionally Directed Medical Research Programs. DOD actually funds more prostate and breast cancer research than NCI does. And in general, they're the second largest funder in the US of cancer after NIH. So the grants we give out is great. The SBR program is wonderful too. However, I would, if you have bandwidth and interest, look into partnering with the federal labs as well. So thank you.
You know, Michael could be a great resource, and I should take the opportunity to get to know him. And Michael, thanks for being here. So other questions or comments? I have one other person that I want to ask a question. And so even though you're going to hear from him later, our St. George winner is not only a great academician, but he's also a great businessman, and he serves on the boards of companies and creating companies and so on and so forth. So Rakesh, do you have any advice to these people thinking about companies?
Raju, they want to hear me this evening anyway, so I think they would rather go home and have a glass of wine or something right now. I think when I sit on the board of Tekla, as you know, there are 4 funds, about $3 billion. And I think the most important thing we look at is CEO, CEO, CEO. And of course, science is the most important thing, but without having a right person, you can give the most wonderful thing to somebody whose heart is not into it, or they are not really that would be my, at least the only thing I've learned in the last 25, 30 years, the CEO, and of course the science behind it. So, thank you, Raju.
Thanks. So before closing, I just want everybody to just to sort of make a crisp statement. What really excites you about the companies that you started? So we start with Vlad at the end.
So what was the question?
What is it about Chimera that so excites you?
It's a strong team, passionate team, great science.
Oh.
I think the opportunity to pick up on an area that seems to have been underexploited and really do something effective with it.
Ken?
That I get to work with you, Raju. Oh, and Chico.
Okay, so I guess the innovative part, the innovation part, the undisclosed, the part of entrepreneurship is always, in a world of innovation, you meet with innovative therapists on development, of course, but almost every day. And for me, it was an innovation also jumping to the business side. So I guess that's it.
Thank you. Pritesh.
For us, North Star is patients, patients, patients. I want to make a difference in the lives of patients.
Wonderful. John?
Yeah, for us, we have a drug with a very high therapeutic potential to treat GI cancers.
Great. Yeah. So I want to close by saying, going back to Suzanne, Suzanne has 3 bosses because she has 3 organizations. Each of these organizations has a board of directors, and they have a chair of the board of directors. And so she has to answer to all of them. And so we heard from Alfred Slanitz earlier, and then we have Charlie Weatherspoon, who is the chairman of the AFCR. Where is Charlie? And then we will hear from Lance Karaguchi, who is the chair of AFCR, and Charlie started as the founding chairman for the AIM High, and he has been helping Suzanne, and he's going to close this session. And we can all go. Thank you. Thank you.
Say, wow, finally, huh? I see some eyes over there that's a little dazed. But what I'm going to give you guys the opportunity, stand up and give our crew a great round of applause. As always, don't sit because I know you're tired, so stand up. This is going to be quick. First of all, as always, thanking Suzanne, but Suzanne is a great leader, but without some great folks helping you, and especially for this particular occasion, looking at Brian, Ella, and Hallie and the great work they're doing, and also for Suzanne's family who come here to support her. So it's very, very important that we know that and understand that. I can't say enough about how far we've come, known Webb and Raju and everyone for a long period of time, but this whole thing has changed to the point where you're making a difference in everyone's lives. And I think the issue of diversity, the culture that's here at NFCR and AIM High is very important because we're changing not just the neighborhoods, we're changing the world. And it's a world situation that we're looking at. That's why I say it's the global summit. It's not the U.S. summit, it's the global summit. And thanking NFCR for supporting AIM High, and we look forward to seeing you at the St. George Awards. Thank you, and have a good day. And for the older folks, take a nap. For the young folks, go to the bars early.